Professor Groner, thank you very much indeed for joining us on eCancer TV, just for 2 or 3 minutes to tell us a little bit about your work in Frankfurt. And you're interested in mouse mammary cancer, and you're interested in what goes on inside the cancers and what's the stem cell component, and is it important or who cares? And you know that this is an interesting issue for me working in the European Institute of Oncology, where we're also very keen on stem cells. Tell us about your work.
Well, we're interested in stem cells in normal tissues and stem cells in cancerous tissues. And the idea is that there is a relationship somewhere between both of them. What we have been doing is we have been identifying stem cells in normal tissues through transplantation experiments. So in the mouse at least, you can replace the epithelial component of the mammary tissue by exogenous cells. So you can clear the fat pad of epithelial cells. You can then transplant epithelial cells into the empty fat pad, and you can see that the stem cells give rise to a normal-looking mammary tissue. And that is a definition of a stem cell.
Sure. And unfortunately the only one at the moment.
It's a functional definition. We say a stem cell can reconstitute an organ.
Yes.
And we have a couple of markers, so molecules which are preferentially expressed on stem cells.
Such as?
These are surface molecules, molecules which can be recognized by particular recombinant monoclonal antibodies. This is CD44, CD49, CD24, so different.
You need all three?
If you combine the selection for all three, you can highly enrich stem cells, but you still cannot identify or purify stem cells. So you know that the stem cells are somewhere in the population, but you cannot really purify them to an extent that you can do a good genetic or biochemical analysis.
So you can't go in with your stains and say, that's the one stem cell, I'll put that in the fat pad and prove that that's the one that differentiates into three tissues.
This mouse system is most amazing, because I can use unselected cells. So you can use the whole population of cells and can see that stem cells are in there. And then you can go down, you can enrich them, and a smaller and smaller number of implanted cells are then able to reconstitute the ducts.
So you do doubling dilutions, essentially.
You do enrichment.
Yes. So what are you going to do now that you've got this system?
Now, we went a step further and genetically modified the stem cells. So you can first take them out of an animal, then do a genetic modification experiment. You infect them with what's called a lentiviral vector. Now, this is a gene transfer vector. So you can introduce genes into the stem cells, or you can knock out genes in these stem cells. And then you can ask which genes are really required in order to maintain the stem cell function or to transform stem cells. And there we made a very interesting observation, and that is when you activate a particular transcription factor, that is STAT5, in these stem cells, and then you reintroduce the stem cells into the fat pad and other limbic tumors. And that makes a very nice correlation with what people observe in human tumors. So if you analyze human tumors, you find that maybe 60-70% of the primary human tumors have an activated STAT5 in it. So here you can extrapolate from an experimental observation in a mouse to a clinical observation in human patients.
And what happens if you knock it out?
if you knock it out, yeah, very nice question. You observe exactly the mirror image. So if you induce the transcription factor, the cells grow more and they're more resistant to apoptosis, and they go faster through the developmental differentiation steps. If you knock it down, then the ducts still form, so they still can go on, but any type of hormone-induced differentiation, so massive proliferation of the epithelial cells during pregnancy, doesn't occur anymore.
Oh, wow. Interesting.
So the transcription factor is required for driving the cells through the differentiation process, protecting the cells from apoptosis. And if the transcription factor is not turned off, as in normal cessation of lactation, then the cells transform and become tumors. And that could be, say, a very nice model for human tumors, simply because the correlation between SET5 activation
and you published this work?
Yes, it's in press.
Whereabouts?
In a journal called Stem Cells.
What happens if you haven't yet got around to looking at it in tumor stem cells, have you?
Well, we have done transplantation experiments with this tumor.
You could knock it out in a tumor cell, in a tumor stem cell.
Yes.
That would be really...
At this point, the model says if you activate it and do not down-regulate it at the proper time in normal differentiation process, then it's bad for the cell, it becomes a tumor cell. And then the other point is that, as I said, you find it in many primary tumors activated. Now, your question would be, what would happen if you down-regulate it in the tumor stem cells? And these are experiments we're about to do.
Yeah, that sounds interesting.